Welcome to Chris Biology. So today we are going to talk about antibodies. So if you've seen my last video, so you will get a continuation of what it means, right? So we are specifically talking about B cell immunity. So if you haven't watched, please go back and watch the 5-minute video on immunity. So going about today's video, antibody is nothing but immunoglobulins. These are nothing but proteins that are secreted by B cells, right? So in later part of the video, I'll tell you how they are produced. But before that, I just want you to know the structure of a immunoglobulin, right? So globulin is nothing but the protein. Globulin is a plasma protein. Immuno is just meant for immunity, right? So it has 2 heavy chains and 2 light chains. That is why it is represented as H2 and L2, right? So it has these heavy chains and light chains are connected by disulfide bonds, as you can see in the picture. So it has one end called FAB and another end called FC, wherein FC end is attached to a mast cell or an antigen presenting cell, which is nothing but a macrophage. We saw it in the classification of innate immunity, right, if you remember. Next, there is something called an epitope and a paratope, right? So what is an epitope? Epitope is a antigen binding site on the antibody. Vice versa, what is a paratope? Paratope is the antibody binding site on the antigen. So you just have to remember one thing. Maybe we have to remember epitope so that we will remember vice versa is paratope, right? So that was about the structure of the antibody. Next, coming to what are the types of antibody, right? So the types of antibody are of 5 types. So you can remember with the mnemonic GAMD, right? GAMDE, GAMD. So IgG, A, M, and DE, right? So highest concentration, plasma concentration, highest percentage is G and lowest is E. That is why I have arranged in that particular order, right? GAMD, that is how you can remember it. So if you look into the structure, not all antibodies are, monomers, right? They are different. So IgG, IgD and E are monomers, while A is a dimer, M is a pentamer, right? So these pentamers or dimers are connected by joining chains, wherein in a pentamer there is only one joining chain and there are 5 disulfide bonds which are connecting the 5 pentamers, right? Next, what if you want to look at the function of what each antibody does, right? So G crosses placenta, if you know. Next, you have something called chronic inflammation. What is the antibody involved in chronic inflammation? It is IgG. If you come to IgA, you already know it is already it's the antibody present in the secretions. I already specified, right? In the secretions, in the acid, in the tears. What is the antibody present? IgA. So it is also present in the breast milk, right? So both these are neat points. Which antibody crosses placenta? IgG. Which antibody is present in breast milk? IgA. What is the function of IgM? IgM plays a role in acute inflammation, right? So acute and chronic both are inflammations only, but acute means a short duration, chronic means a long duration. IgD, not a specific function, but it has a receptor like function, not very sure. IgE plays a very important role in allergic response in people who are having asthma or any sort of allergy, right? So there, which antibody plays a role? It is IgE. Remember this, it's very important. Next, what is the next question? It's about how antibodies are produced, right? How are they produced? So what happens first is, if you remember from my last class, I have specifically told both antigens and antibody, whatever is interacting is also dependent on T cell, right? It's not wholesale B cell function, right? It works in a harmony, right? So what harmony will it work in? There will be an antigen, there will be an antigen presenting cell which will attach the antigen or will get to know about the antigen. Then it will send signals or some cytokines to T helper cells. T helper cells will further help in the recognition and in the activation of B cells. Now these B cells, after activation or after getting signals from T cell, will get converted into something called a plasma cell and a memory B cell, right? Memory B cell is for your immunity to remember that this antigen was already processed. Plasma cell will produce antibodies. What is the function of an antibody? it will either neutralize the antigen or it will do phagocytosis. It will give signals to the macrophage for engulfment of the antigen, which is called phagocytosis, or it will do a complement mediated lysis. There are something called blood complements, right, which are again proteins present in the plasma. This will lyse the antigen, or it can do an antibody dependent cellular cytotoxicity. Like we said, natural killer cells make holes. The same way antibodies have a role in cytotoxicity. So once the cytotoxicity is done, it is going to kill the cells, right? So I hope this was a crisp thing about antibodies. So if you like my videos, please do subscribe and share. Thank you.